Welcome to the hopefully 10 minutes long suggested talk. My name is Thomas. I'm working at the Botany 1 in the group of Peter, Nick, and my PhD project currently is called genetic food diagnostic. Last year in spring, my PhD thesis started with the initiation of a cooperation between the company Phytocom and our institute. The company is called Phytocom and it imports traditional Chinese medicine pharmaceuticals in the form of granules, and these granules are made from raw drugs. Most of them are from plant material, plant parts, and others are also from a few animals and some inorganic drugs. Here's a picture of some boxes with granules, and these granules are made from starch, cellulose, and maltodextrin as the carrier matrix, and an extraction product from these pharmaceutical plants are transferred to this matrix. The company is of course interested in if there's DNA still inside these granules, and for me the question then is how to make a diagnosis to distinguish between these granules and their components. And for the company this is of interest because of authentication. Each of these products has a name and is made from should be made from a specific plant according to traditional Chinese medicine, and so we are trying to prove that with the DNA that the specific plant is inside or has been used to produce these granules. There's also quality and safety, which is important for somebody who is selling these granules, and especially with medicinal plant products it's highly important that the correct species is been used for these granules. So to distinguish, first we should know something about a product, and not necessarily something about traditional Chinese medicine, but at least something about the background. And so there's a big book called Materia Medica from Dan Bensky, and in this book these different pharmaceuticals are described, and here's one example. It's called Dangui or Angelica sinensis radix, and this pharmaceutical has one standard species, it's Angelica sinensis, and there's also a substitute species, Angelica acutiloba, and an adulterant. So for each of these hundreds of different pharmaceuticals there are one or more standard species that are mainly used for these products, and there are sometimes also a lot of substitutes which can be used but are not that good in quality, and there are also quite often adulterants known which shouldn't be used for this product because it has a maybe different effect, no effect at all, or it's toxic even. Now to distinguish, there are two scenarios. First, this little citation from Wikipedia: genetics is the science of genes, heredity and variation in living organisms. So to make genetic food diagnostic and to distinguish between different plants that should be used or should not be used for a certain product. There are two scenarios. One is for these specific plants, sequence data is already available, but this part comes later, so I choose the second scenario first. It's if there's no sequence data available for the plants you want to distinguish genetically. Then we have to find variation, and my research showed that pattern generation is one of the approaches to find variation in the genome of organisms, and there are two ways or two methodologies. One is by creating restriction fragments, and the other is by just creating amplification fragments. In the following, I kind of want to show you 3 examples for this pattern generation. The first one is called restriction fragment length polymorphism. You digest genomic DNA, and so you get a fragment pattern of the cutting sites, and you can compare organisms according to these patterns. An more advanced technique is amplified fragment length polymorphism, which uses the restriction digestion in addition together with PCR technology, and it's working like this. You digest genomic DNA with restriction enzymes, then you get fragments which have a known sequence at the end because you know where the enzymes cut. To these known ends you ligate an adapter, and this adapter is also a known sequence for you, so you can use the adapter for primer design, to use primers, and you can amplify from a big pool of fragments from different size. So you can amplify a specific group by extending the primer, so this extension reaches into the unknown of the fragment, and by doing that you get these fingerprints which you can see on the right side, and by choosing more and more extension you get a clearer picture of the fingerprint, less fragments. The third and last method is random amplified polymorphic DNA. In this case you just use short primers and make a PCR reaction with one primer. You generate a fragment pattern again by amplifying certain fragments that are bordered by these sequence of the primer, and yeah, it is a rather bad method to make authentication because it's not good to reproduce the results. In my experience, even the concentration of the template changes the pattern, so it's not very stable fingerprinting. Okay, now we have visual distinguishment. No, we have visual data of variation, and we want to get sequence data from this visual data. How can we get that? We did I choose rapid RAPD, random amplified polymorphic DNA method, to make these fingerprints or these fragment patterns of 8 different plants, and one of these fragments was chosen because it's unique between the plants. The fragment was isolated, re-amplified, and the fragment was sent to sequencing. It was sequenced, and with the sequence data you can design primers, specific primers hopefully, and if you test them you get either such a nice picture where only in the one plant you want to identify a fragment is created and in the others not. So this would be a diagnostic primer to be able to say that in a certain product you have this plant or DNA from the plant in the product. Okay, to finish all what I started, genetic barcoding is also a method to authenticate products, to identify and distinguish between organisms. In this case, you need sequence data that's already available. Best would be if already a barcode existed. For that, someone would have to acquired specimens and also determined what kind of species it is on morphological ways. Then a certain region of this organism is amplified and sequenced, and it is saved in a database and would be then the barcode for this certain species. If this data is available in a database, we just would have to amplify this region with our sample and check the sequence data with the database. That's the principle of barcoding. The problem is that in the plants there is no fixed barcode region. There are some suggested regions, RBCL and MATK, but they seem not to be able to distinguish all plants. You have to kind of analyze the variation of some segments, and the variation shouldn't be too big inside a species, and between species it should be enough to be able to distinguish between different species. So to come to an end, this is what I've done so far is very applied. I try out a lot of extraction protocols for DNA, different stuff. So, but there's no real, okay, it is science, but it's not that science. So a less applied future for me would be to make variation analysis or barcoding analysis with some of the medicinal plant genera I'm using. Also, one could test effects of medicinal components from these plants on plant cells. Also, I would like to extend the TCM plant collection of the botanical garden, or I want to help with that. And maybe I can continue to work at high throughput automated sequence analysis, which I've done. So this is it for today. Thank you very much. Oh, sorry.